3 billion years ago, light first zipped through chlorophyll within tiny reaction centers, the first step plants and photosynthetic bacteria take to convert light into food. Heliobacteria, a type of bacteria that uses photosynthesis to generate energy, has reaction centers thought to be similar to those of the common ancestors for all photosynthetic organisms. Now, a University of Michigan team has determined the first steps in converting light into energy for this bacterium. Our study highlights the different ways in which nature has made use of the basic reaction center architecture that emerged over 3 billion years ago, said lead author and physicist Jennifer Ogilvy. We want to ultimately understand how energy moves through the system and ends up creating what we call the charge-separated state. This state is the battery that drives the engine of photosynthesis. Laser 2. JPEG photosynthetic organisms contain antenna proteins that are packed with pigment mole. The collected energy is then directed to reaction centers that power the initial steps that convert light energy into food for the organism. These initial steps happen on incredibly fast timescales, femtoseconds, or 1/1,000,000 of 1/1000,000 of a second. During the blink of an eye, this conversion happens many quadrillions of times. Researchers are interested in understanding how this transformation takes place. It gives us a better understanding of how plants and photosynthetic organisms convert light into nourishing energy. It also gives researchers a better understanding of how photovoltaics work, and the basis for understanding how to build them better. When light hits a photosynthetic organism, pigments within the antenna gather photons and direct the energy toward the reaction center. In the reaction center, the energy bumps an electron to a higher energy level, from which it moves to a new location, leaving behind a positive charge. This is called a charge separation. This process happens differently based on the structure of the reaction center in which it occurs. In the reaction centers of plants and most photosynthetic organisms, the pigments that orchestrate charge separation absorb similar colors of light, making it difficult to visualize charge separation. Using the heliobacteria, the researchers identified which pigments initially donate the electron after they're excited by a photon, and which pigments accept the electron. Heliobacteria is a good model to examine, Ogilvy said, because their reaction centers have a mixture of chlorophyll and bacteria chlorophyll, which means that these different pigments absorb different colors of lights. For example, she said, imagine trying to follow a person in a crowd, but everyone is wearing blue jackets. You're watching from a distance and you can only take snapshots of the person moving through the crowd. But if the person you were watching was wearing a red jacket, you could follow them much more easily. This system is kind of like that. It has distinct markers, said Ogilvy, professor of physics, biophysics, and macromolecular science and engineering previously. Heliobacteria were difficult to understand because its reaction center structure was unknown. Thanks for watching. Please subscribe my channel for more updates.